Okay, so let's go over bacterial cell structures, and I've already started by drawing a shape of a random made-up bacterial cell. It's not any particular type of bacterium, just something I made up. And the first thing that our bacterial cell has to have is a cell membrane. And the function of a cell membrane is to define the boundaries of the cell. It controls what goes in and out of the cell. And in bacteria, the cell membrane is also the site of energy synthesis or ATP synthesis. The structure of a bacterial cell membrane is almost identical to the structure of a eukaryote or human cell membrane. That is, the cell membrane is a phospholipid bilayer. Now, this is going to be important later on in the semester when we talk about antibiotics and how they target bacterial cells, but not our cells. We'll learn that there aren't many drugs that affect bacterial cell membranes, because if they attack the bacterial cell membrane, that would mean that drug also attacked our cell membrane, and that's not a situation that we want. So we're not going to talk a whole lot about the structure because it should be review. It is similar to your cell membranes, but the similarity between those two is a very important thing that we'll talk about later on in the semester. Now, if we move inside our little bacterial cell, we have a single circular piece of DNA, and this is the chromosome. And it is made of DNA, and this contains the genes of the cell, or all of the directions that the cell needs to carry out its daily living, its metabolism, how it reproduces, and so on. So one single circular molecule of DNA. Unlike us, we have 46 linear molecules of DNA, or 23 pairs. In a bacterial cell, there are no membrane-bound organelles. So the DNA is not in the nucleus because there is no nucleus. Instead, that DNA is found in an area or region. We call it the nucleoid region. It's not defined by a boundary. It's just an area where the cytoplasm gets a little bit thicker, and it's where the chromosome is. And it's not called a nucleus, of course. It's called a nucleoid, which means nucleus-like, and it's where the DNA is located in the cell. Now, bacteria are really cool about some of their structures and their functions and the things they can do. One of the things that they can have is called a plasmid. And a plasmid is a small piece of extrachromal DNA. By extrachromosomal, I mean it's not part of the chromosome. And these are like bonus genes. The bacterial cell doesn't need the plasmid to survive, but the plasmid gives it some extra capabilities. And we'll talk about this more later on too. Plasmids are really nasty as far as humans are concerned, because sometimes the antibiotic resistance genes, the things that allow them to be resistant to our treatments, those genes are located on these plasmids. And the bacteria have mechanisms for sharing these plasmids amongst each other. Also inside the bacterial cell is a structure that you probably recognize, and that is the ribosome. And the ribosome has a small subunit and a large subunit, and hopefully you remember that the ribosome is the site of protein synthesis, very similar to how it works in a human. And then, of course, inside this bacterial cell, we have the cytoplasm, which is the contents of that bacterial cell. Now, moving just to the outside of the cell membrane, we have a very important structure. This is one of the structures that we're going to talk about today, and we're going to talk about throughout the rest of the semester because it plays a huge role in how bacteria respond to antibiotics. It plays a role in how the immune system attacks different bacteria. It plays a role in the body's response to these bacteria, because in some bacteria this structure is toxic, and others it's not. So we're going to come back to this structure again and again throughout the semester. So it's important that you learn as much as you can about it early on, and that structure is the cell wall. And the bacterial cell wall is made of something called peptidoglycan. And yes, you do have to learn how to spell peptidoglycan. And it is a unique substance found only in bacteria. As far as we know, no other types of organisms contain peptidoglycan. It's unique to bacteria. Now, peptidoglycan is made up of alternating subunits of N-acetylmuramic acid and N-acetylglucosamine. And we're just going to abbreviate those things, NAM and NAG, just because it's easier and we don't have to write N-acetylmuramic acid and acetylglucosamine all of the time. And the N-acetylmuramic acid and N-acetylglucosamine, NAM and NAG, are strung together like beads on a string. So if I just draw this, I'm going to make my NAM subunits blue, and I'm going to make my NAG subunits red, and draw these. And it looks like maybe a little kid was making a necklace and alternating different colors of beads on this string. This one got a little funky. And so they are linked to each other with chemical bonds. But what makes peptidoglycan special, the important thing about peptidoglycan that you need to remember, is that the NAM subunits cross-link to each other with these glycan bonds. So these cross-links between the NAM subunits make the peptidoglycan very, very strong. Otherwise, it would be like wrapping a ball with a piece of twine. The twine would continuously slip off. These cross-links make the peptidoglycan more like chainmail armor. It gives it a lot of strength so that the bacteria can resist bursting due to osmotic pressure. So the one thing the cell wall does is it makes the cell wall strong. The other thing this does is it gives the cell its characteristic shape. Now, cell walls come in two varieties. There are gram-positive cell walls and gram-negative cell walls. Now, these are both named for the scientists who created a staining procedure which allows us to determine whether the cell is gram-positive or gram-negative, and his name was Hans Christian Gram. And so we always capitalize gram whenever we write it because it is a proper name. So if I were to differentiate between a gram-positive cell wall and a gram-negative cell wall, first I'm going to draw my cell membranes. Now, the cell membrane is not part of the cell wall. So immediately to the outside of the cell membrane in a gram-positive cell, I have a thick layer of peptidoglycan. It's a very, very thick layer. Sometimes in a gram-positive cell, the cell wall can be 30% of the dry weight of the cell. So it's a very thick. So gram-positive cells have thick peptidoglycan. And then the gram-positive cell also has these acids associated with it. These lipoteichoic acids anchor the peptidoglycan to the cell membrane, and then these teichoic acids stick off the surface of the peptidoglycan and are used for things like cell-to-cell recognition and communication. So the gram-positive cell has a thick peptidoglycan with these teichoic, and yes, you need to know how to spell this as well, and lipoteichoic acids. If we go over here to our gram-negative cell wall, we have our cell membrane, and then to the outside of the cell membrane begins our cell wall structure. It has a thin layer of peptidoglycan. And to the outside of the peptidoglycan is another membrane now. We do not call this a cell membrane. It's not a second cell membrane. It's called the outer membrane, and it's part of the cell wall. So these two things, the peptidoglycan and the outer membrane, make up the cell wall of a gram-negative cell. Now this outer membrane is unique. If we just kind of make a little blow up here, it's not a phospholipid bilayer. The inner leaflet, which is the part closest to the peptidoglycan, is made up of phospholipids. Just like the cell membrane. But the outer leaflet, the part that's exposed to the exterior environment, is made of something called lipopolysaccharide. Or LPS. And so this outer membrane contains LPS. So if we compare the gram-positive and the gram-negative cell, what we would say about the gram-positive is it has a thick layer of peptidoglycan with teichoic and lipoteichoic acids and no outer membrane. And the gram-negative cell has a thin layer of peptidoglycan with an outer membrane that contains LPS. Now, hopefully at this point you're asking yourselves why this is important. So the reason we need to know if something is gram-positive or gram-negative, reason number one is for antibiotics, prescribing the appropriate antibiotics. Some antibiotics work by disrupting the integrity of the peptidoglycan, causing the cell to rupture and die. Those kinds of antibiotics tend to work better against gram-positive cells because the antibiotics have access to the peptidoglycan. With a gram-negative cell, those antibiotics can't access the peptidoglycan because they're blocked by the outer membrane. They can't get to the peptidoglycan to destroy it. One type of antibiotic that works by interfering with that peptidoglycan is penicillin. Another reason we need to know if something's gram-negative or gram-positive is because this LPS is actually quite toxic to humans. And so if somebody is sick, they have a bacterial infection, we need to know if it's gram-positive or gram-negative so that we know if they're going to be susceptible to septic shock induced by LPS. The LPS somehow triggers our immune system to go into overdrive, and this causes septic shock, and often a person who has a gram-negative infection will die of organ failure due to septic shock. So we need to know what kind of bacterium we are working with so that we can prescribe antibiotics appropriately and we can monitor for signs of septic shock. One of the things we'll do in lab so that we can quickly determine if something is gram-negative or gram-positive is a Gram stain. That's the most widely used diagnostic procedure in microbiology, is the Gram stain. And we will do that in the laboratory. Now, if we go back to our little bacterial cell, and we're continuing our journey towards the outside environment. So sometimes bacteria will have a structure located immediately outside the cell wall, and this is called the glycocalyx. And the glycocalyx is made of polysaccharides, and it's very sticky, and it also prevents the cell from drying out. So its functions are for attachment to surfaces. One notable example of a glycocalyx producing bacterium is Streptococcus mutans. It allows the bacterium to attach to our teeth, where it then secretes acidic byproducts and causes cavities. And it also protects the bacteria from the white blood cells of the immune system. Now, with our glycocalyx, if it's thin, we call it a slime layer. And if it's thick, we call it a capsule. Continuing our journey towards the outside of our bacterial cell, sometimes there are protein projections from the surface of the cell. There can be long ones and there can be short ones. And the long ones are called pili, and the short ones are called fimbriae. And these are for attachment. And our final cell structure is one you're probably familiar with. These long things are the flagella, and they are for motility or movement. Now, something that's important to know about this bacterial cell that I've drawn, this is an imaginary bacterial cell. Not all bacteria have all of these structures. All bacteria will have a cell membrane. They all have ribosomes. They all have to have a chromosome. They all have to have a nucleoid region and a cytoplasm. Now, most bacteria also have a cell wall, to the point where most microbiologists say all bacteria have a cell wall. So when I ask you the structures that all bacteria have, I want you to say they all have a cell membrane, cytoplasm, chromosome, nucleoid, ribosomes, and a cell wall. Now, as far as the cell wall thing goes, though, there is one genus that's a notable exception. And because there's only one genus, that's why the microbiologists kind of say all bacteria have a cell wall. And that genus is called Mycoplasma. This particular genus has no cell wall. And this is the bacterium that's responsible for causing walking pneumonia. So it's the only genus that doesn't have a cell wall. So when I say, what structures do all bacteria have? They have a cell membrane, a cell wall, ribosomes, cytoplasm, chromosome, and a nucleoid. These other structures are dependent on what kind of bacterium we're dealing with. For example, E. coli will have flagella, but they don't have capsules. Streptococcus mutans, that causes cavities, has a capsule, but it doesn't have flagella, and so on. So the remaining structures kind of mix and match dependent on what type of bacterium we're dealing with.